All right. So compared to all the previous speakers, I'm not a neurologist and I'm not even a clinician. So I'm a cell biologist, and my main interest is telomerase, together with telomeres and also aging in senescence. However, we got interested in Parkinson's disease and neurodegeneration in general, and I will try, let me just move this. I will try to show you the data we have. So these are mainly mouse and cell studies, so not many patients involved. So for those of you, I don't know whether I can remove this. Don't know whether you can, whether you can see the headings. So there's something on top of it, but I don't want to move it now. So for those of you who are not so aware what telomerase is, it's a reverse transcriptase, and it's normally there to maintain telomeres. So the telomere are at the end of chromosomes. And so the important part is the protein part, which is called TERT for telomerase reverse transcriptase. So I will use this abbreviation quite a bit. But in order to provide a template region for the synthesis of telomeres, there's an inherent RNA part, which is called TERT for telomerase reverse component. However, as I already mentioned, these two components are important for the elongation of telomeres, which is called the canonical function. However, during the last rather 20 years, it became more obvious that there are also non-canonical functions. So, and I will focus on these additional functions, which normally relate only to the TERT protein. So what we found in what we published in 2008 is that if you use an antibody against the TERT protein in cells, like these are normal fibroblasts, which overexpress the H-TERT antibody because the H-TERT protein, because normally those fibroblasts, like most normal human somatic cells, don't have the telomerase activity and also an active TERT protein. So in order to perform its canonical function, because all the chromosomes and the telomeres are naturally in the nucleus, the protein to maintain telomeres has to be in the nucleus. However, if you now apply a stress, like for example, an acute stress, H2O2, or a more physiological stress, when we have hyperoxia in the incubator where we grow the cells, and instead of the ambient 21% oxygen, you have now 40%. Just check. Yeah, time. Okay. So, and while it's very quick, if you apply 3 hours of either 300 or 500 micromolar of hydrogen peroxide, you can appreciate that the protein, which previously was in the nucleus, either partially or completely leaves the nucleus. And yeah, it takes much longer under hyperoxia, which is more physiological, but you see after 10 weeks, we have the same situation like after 500 micromolar H2O2. And if we now use a second color, the mitotracker, which is green, while TERT protein is shown with a red secondary antibody, you can now see that we have also got quite a number of yellow signals. And it's not only our observation, but other people found a similar thing that the TERT protein under a stress condition can go into the mitochondria. So this is clearly a role or a function, if you want, that is not directly or not even indirectly connected to telomere maintenance, because the mitochondria have a circular genome. So if you look now to the phenotype, what it does there when it is more in the mitochondria. So we now compare here in black the parental fibroblasts, which don't have the TERT protein, with red ones, which are H2 overexpressing, and we measure the oxidative stress. So first we have the mitochondrial superoxide using a mitoscope. It is already decreased under normal condition, and it is also decreased even more if we look for all intracellular peroxide in the cell under hyperoxia. And if we look now to the integrity of the mitochondrial DNA under conditions of either acute H2O2 treatment or again the hyperoxic conditions we have. So here now in black are the normal fibroblasts, and open circles and dotted line are the MSCH TERT. So you see that we have much less mitochondrial damage. And here we think that even the time, this is the time where we have the majority. So it's now 40 days. It's not exactly 10 weeks, so it's here, but it's already a large majority of the TERT protein in the mitochondria, which seems to protect it. And if we look to apoptosis, which is of course also related to mitochondria, either H2O2 treatment again decreases the sensitivity of the cells to the agent and decreases apoptosis, and we see an even larger effect if we use etoposide, which is used, for example, as a therapeuticum for breast cancer treatment. So then I mean these experiments were all done in cell culture. So we were now interested, does it play a role in brain in vivo? And the experiments we did for this is we cultured primary mouse neurons, and after a while, I mean, they are initially positive for telomerase activity, so it's measured here as absorbance, but after 2 weeks time, or yeah, a bit more than 1.5 weeks, telomerase activity decreases, but I show you in the next slides that the TERT protein still is there. The mouse brain is a little bit different. Here you can have telomerase activity in the brain until 3 weeks postnatally. However, in humans, the situation is completely different. Here you see post-conception weeks. So these are done in brains from aborted fetuses. And you can see that after 6 weeks after conception, there's still a very reasonable amount of telomerase activity, but it decreases sharply, and essentially after week 14, you barely have any telomerase. So I already told you that telomerase activity is one thing, but we looked for the protein, and this is now human tissue, so it's human hippocampus. First, we looked with immunohistochemistry, and you see the brown staining of neurons for TERT, or we have now the green secondary antibody, and we co-localize this Neuro trace in order to make sure we only look to neurons. And again, it seems that neurons have still persisting TERT. We have a little bit in microglia because those are macrophages, and they are known to be positive for both TERT and telomerase activity. But importantly, we don't have anything in astrocytes. So now here the TERT protein is in red in those two cases, and there's no co-localization with GFAP, which is marker for astrocytes. And if you look for into cultured mouse neurons, again, TERT protein in red, we see co-localization with neurons, with microglia, but again, nothing with astrocytes, and we also don't see anything in TERT knockout brains, which is important because a lot of antibodies are not working very specifically. So having now shown that there is indeed some TERT protein without telomerase in human brains, and I have to admit that these are all very old samples, because I will show you the study was designed to analyze Alzheimer's disease patients, and we used hippocampi from controls, which we used here, which are age matched, and so they are at least 70 to 80 years old. So yeah, so first I show you the study we have done in Alzheimer's disease brains, and then in the second part, what we did as a mouse model for Parkinson's disease. So just to summarize quickly our paper, which was published in 2015, we assumed, as we know, in mouse brain, the telomere, the TERT protein decreases during aging. We didn't see, so we thought not only during aging, but maybe also during disease, but we didn't find this, and I don't show it here. However, what we found is more TERT protein localized in the mitochondria. So you remember what I initially told you about our cell culture experiments. So it's known that oxidative stress occurs not only in old brains, but especially in brains from people who suffer from neurodegeneration. So let it be Alzheimer's disease or Parkinson's disease. And we found all three CA1, 2, and 3 regions. The Bragg stage 6, which are the highest stages of Alzheimer's disease, have significantly more TERT protein in the mitochondria than age-matched controls, which are shown in white bars. However, what we don't know exactly is what it means. So intriguingly, when we have Bragg stage 0, we only see TERT protein shown in red in the neurons. There's no pathological tau, which we detected with the 88 antibody. However, if you look into higher Bragg stages, so starting from 3 until 6, you see the neurofibrillar tangles or threads shown here in green. And what we found is that there's never a co-localization with TERT. So there are two possibilities. Either those cells which have a lot of TERT never develop those neurofibrillar tangles of tau protein, of pathological tau protein, or once the NFTs are formed, then the TERT protein is displaced from the neurons. Since we only had those end-stage brains and you can't do any intervention with those, we decided to use the cultivated mouse neurons, and we used them either from wild-type mice, shown in blue, or from TERT knockout mice, shown in red. And if we look now and we transduced them with mutated tau, so we used P301L, and we found substantially more oxidative stress in the dendrites. And we also looked to other markers of oxidative stress, like peroxynitrites with an 4-HNE antibody. And here we found it now that is more in the soma, probably because there are more membranes in the soma than in the dendrites. So then we thought, okay, if there is a protective effect of the TERT protein, as we saw in the wild type compared to the knockout neurons, then why don't we interfere with it? So at this time, it was already known that there are different telomerase activators. So ours are from the American company TA Sciences. And one is a highly enriched plant extract from the Mongolian milk batch, which you can actually buy online, but it's rather expensive. And there's another one which is based on the main component of this plant extract, which is cycloestrogenol, and it's called GRN 510. So what we did initially is we did a pilot study where we treated 2-year-old mice. They were all female mice. Sorry. So first, I want to show you the data which I mentioned previously, that in mice, in brains compared, so old mice, 21 to 30 months old compared to 4 months old, have a less TERT expression. And if we now use these two activators, which I just described in the previous slide, and we treat those 2-year-old mice, we see a significant increase in the TERT expression levels of both activator treated brains compared to the controls, which are treated with DMSO, because both components are dissolved in DMSO. If you now do a rather crude test, because these old mice are really old and you can't do a lot with them, we set them on a static rod, and while for 1 minute for 5 times, and while of course young mice are staying on the rod without problems, old mice, the controls, which were only treated with Sasoli and DMSO, fall down very quickly, so only half of the time they could stay on. But the telomerase activator treated mice were well able, almost like young mice, to stay on, and we got significance for the GRN 510, but we didn't have a lot of mice. So in order to make sure, because brain are brain and contain a lot of different cell types, as you know, and as I showed you before, we cultured again neurons and treated them for 48 hours. And both the GRN 510 and the TA65, the plant extract, were able to increase TERT expression significantly. So it really looks as if it is the neurons. We didn't do any other cell types because it's not really easy to do, to cultivate them. So, I mean, to this audience, I probably don't have to explain about Parkinson's disease, just to remind you that the alpha-synuclein aggregates, also called Lewy bodies, accumulate in the brain. And the main feature, of course, the functional feature, is that there is a disturbance in the dopamine metabolism due to the death of dopaminergic neurons. And this, of course, as well known, results in a lot of movement disorders like tremor, Parkinson, also dementia is possible. And of course, there are also lots of non-movement disorders, but I will focus on those in the ongoing study. And sorry, it should be mitochondrial. And it's also known that mitochondrial autophagy dysfunction can play a role for the disease. So what we did, we used a transgenic mouse model, which was generated by Elizabeth Maslaya in 2000. And here you see the transgenic mice, which have almost identical inclusions to Lewy body aggregates in Parkinson's patients, and the highest expression of the wild type human transgenic alpha-synuclein is in the neocortex and the olfactory region and hippocampus. And interestingly, if you now follow these mice, you can see you need almost 1 year until the amount of the transgene, the human wild type alpha-synuclein, increases in the hippocampus as well as in the striatum, and also just to make sure that really we also see the same phenotype as in human Parkinson's disease. These transgenic mice have less dopaminergic or less dopamine levels, and also less tyrosine hydroxylase activity, which is an enzyme involved in dopamine metabolism. So now we have this model. We started treating them at 4 months because we were not exactly sure when the first phenotypes, so tremor and other features appear. So we treated them until the mice were 18 months, so for 14 months, orally every day. So they got a kind of food mesh instead of pellets, which was enriched with the components. We also tried if we could, and had enough numbers to discriminate between sexes. So we have female mice, and we got a significant increase in TERT expression with both activators. For the GRN, we only, for the males, we found it only with the synthetic compound. However, we had only 3 mice with the TA67. However, if we combine both sexes, of course, we see highly significant increase in the amount of TERT expression. So this was published last year in Progress of Neurology. So we then went on to characterize some of the motor and balance phenotypes. So we used a rotor rod. So we placed the mice on the rod, and now it's not a static rod, but it is incrementally increasing the speed. So you can measure speed, how long the mouse stays on the rod, and the distance it traveled. And interestingly, I showed it here in the different color coding. In the females, we found a significant improvement. So the mice stayed longer on for higher speeds with the natural telomerase activator, while in males, there was no effect with the natural, or almost no effect. And the significance was only with the synthetic activator. So we don't really understand this, but on the other hand, it's known that brain from male and female, like women and men, can be different, and also apparently it has an implication of the effect of the both activators. So in other tests here, we just measured walking speed in order to have a readout for bradykinesia. In the female, again, the TA65 was more significant. However, in the male, we now got a decrease. So the mice run faster with a synthetic activator. And actually, there was also a good trend for the synthetic activators in females as well. So then we did a stride length test, which is very similar to a gait test, which is done in humans, highly sophisticated, with electrodes, where people step on and then the computer measures the gait. We did a very low maintenance method. So we painted the hind legs of the mice with colors, the right with blue, the left with red. And you can already see it's not only the length of the stride, but also the variability. So sometimes you have a very long step, then you have a very short one. And if you now look to the activator treated mice, you see it's quite even, and the length also in general is higher. So here we measured the stride length in the females at the left, for the left, and for the right foot. And then, so both are longer significantly with both activators. And then also the variation, which decreases because the mice walk more evenly. So in this test, curiously, we didn't find any differences in gender. So we have here the males, again, increase in stride length for the left and for the right foot, and also variation decreases. So this means in this test, on the stride length and on the gait, both activators have similar effects. So since we couldn't do other more sophisticated analysis of cognitive functions due to the locomotor or compromise, otherwise we would have done things like bounce maze. We used a novel object recognition, where the mouse first has two similar objects and then a similar object and a novel object. And you assume that a more healthy mouse with better cognitive capabilities would be more interested in the novel object. And that's what we find for the females with both activators. However, again, for the males, only the GRN 510 had an effect. So since you remember in the very beginning, we were interested in the mitochondrial localization of the TERT proteins. So we also looked for this here. We didn't discriminate between sexes here. So we isolated the mitochondria and did an amplex red test, which measures the H2O2 release from those isolated mitochondria. And we measure electron flow, where we find now TA65 has an effect, and the reverse electron flow using different substrates for the mitochondrial respiration. And interestingly, here we only find TA65. So we think that the mitochondrial effect is not the underlying mechanism for the beneficial effects we saw in the movement and in the gait experiments, because we often see beneficial effects of the GRN 510, but we don't see it here. So we looked for other mechanisms. But first, what we did after we sacrificed those animals, we looked into the amounts of alpha-synuclein. So the green here is total alpha-synuclein. It's in CA1 of the hippocampus. The phosphorylated is shown in red. And here we see the controls where you have quite a lot. So this is merged between red and green. And this I have to emphasize that this is all the same exposure in the microscope. So we kept this very constant. However, you see the treatment with the synthetic compound decreases both the phosphorylated and the total alpha-synuclein levels. And you see with TA65, you barely have any phosphorylated, and you have a little bit from the total alpha-synuclein left. So the next slide is a bit busy. Quantifies this already in the 3 regions. We focused on the hippocampal region CA1, CA3, and also the neocortex, because remember that the transgenic human wild-type alpha-synuclein is most expressed in these areas. And we see in the CA1 all 3 or both the total related to the DAPI staining and the phosphorylated also to the DAPI staining. But then we said if you have less total, you can also phosphorylate less. So we also got a ratio between the total and the phosphorylated. No, it's actually phosphorylated to total, sorry. So there is less, even with this ratio, you still have specifically less phosphorylated alpha-synuclein, even if you have less total alpha-synuclein, and this effect is highly significant, mainly in TA65. For the other brain regions, we have mainly the effects on total, a little bit on phosphorylated, but not in all regions to the same extent. Also, when we take the phosphorylated ratio related to the total. So, because some of our pathologist colleagues said, yeah, maybe it's an artifact of fluorescence, we did the classical staining with a visible substrate, which is here in brown. But again, you can see we now analyze the layer of pyramidal neurons in the CA1. You have a lot of brown dots. It gets less with the GRN 510, and you have hardly any with TA65. And this is quantified here. And we analyzed exactly the same layer because you can't analyze the other layers with immunofluorescent, and you see it matches very well. So that means independent of the method you're applying, we see the same specific effect, that the telomerase activators decrease the amount of alpha-synuclein. Of course, even more important than total and phosphorylated alpha-synuclein is the aggregated alpha-synuclein, which is now shown here in red fluorescence color. And again, as a control, we have the total. And then we look onto how many of the total alpha-synuclein are also red. And again, you can see not only the amount of total alpha-synuclein decreases, but also the amount of colocalization. So that means the little bit of total alpha-synuclein which is left after treatment with the activators gets less aggregated. And you can, again, we quantified it here in the CA1, in the CA2, in CA1 region. Now we have the most significant effect with TA65, but you see we have an outlier mouse here which happens, but in general we also have similar effects with the synthetic one. In CA3 both are significant, and so are we have a high significance in the neocortex, and that's essentially what it's shown. So this is almost my last slide, so I'm coming to the end. So what we assumed is, you remember that it is known that protein degradation mechanisms like proteasome and also autophagy are there in the brain, and they digest different forms of alpha-synuclein. So what we did is we analyzed both the adapter protein of autophagy called P62 and the LC3B, which is sitting on the membrane of the autophagosomes. And you can see both are decreased, the P62 and the LC3, but we have the most significant effect in the CA1. There is quite a striking tendency in the CA3, but less so in the neocortex. However, the P62 is rather well, has a very good trend as well. So we think it could be possible because with age, the functionality of the protein control, including macroautophagy, is decreased in the brains, and that might be a reason why you don't see increased alpha-synuclein or tau or whatever in humans. Neither we saw it in the mouse model until they were 1 year old. So in summary, I will keep it short. I hope I could convince you that a treatment or with telomerase activators in a mouse model of alpha-synuclein transgenic wild-type human alpha-synuclein, those activators improved movement, balance, and gait. And of course, there was also higher expression of the telomerase protein PET. And that this treatment, importantly, on the neuropathological side, it decreased total phosphorylated and aggregated alpha-synuclein. And we think that it's not so much related to any function in mitochondria, but we think that the TERT rather activates protein degradation like autophagy. And there were some papers who showed this in cancer cells, both for autophagy and for proteasome. So if you overexpress TERT, then these protein degradation mechanisms get activated, which is of course not in the brain, but at least there are hints that this is not just speculation out of the blue. So I come to the acknowledgement. Essentially, my Chinese PhD student, Teng Fei Wan, did most of this work, was a little bit help from a master student in the UK, Emma Veer. Previous students, Shahida Ahmed, Alison Spitzberg, Abbas Ishak, contributed some of the background data. And I'm, of course, grateful to TA Science, who supplied us with the telomerase activators, my collaboration partner for the autophagy, Viktor Korochuk, and Johannes Adams, Mary Johnson for the AD project. And we have got a little bit funding in the UK. Thank you for your attention. I hope I wasn't too long.
Thank you for your presentation, Doc. So floor is open for the questions, if anyone have. Any questions from the floor? Doc, I can't able to see any questions. So thank you so much for your presentation.
Thank you very much.